and Maheen, and we can go ahead and kick things off. And I'll make sure to let anyone into the meeting who joins next. But we are recording.
Beautiful. So, as so many of you know, that we have been recipients of the Chan Zuckerberg Award, and I know you don't like this for me to call you Dr. Goebel, but Sarah worked with our wonderful, really the start of Star Foundation, Dr. Stephen Walkley, and she was his student at Einstein, and we are so happy now to be able to have. To bring her into Star Foundation in a more transformative role. So I'm so glad that she's here and really working on behalf of our community and the patients that we serve. And Maheen, you now have been at the NIH for 4 or 5 years.
Yeah, I've
Yeah,
been here 4 years, been working on FSASD for a little over three. So I just
yeah,
really excited to meet everyone and tell you about our work. And people are
yeah.
much nicer than the mice.
I want to hear all about the mice, but I remember Maheen, you being there at their last in-person meeting, and just how what a light you were, and just so encouraging and so excited about our work. And I'm just thrilled to have the both of you on today, and just kind of guiding us through what we can expect and questions that we have, because Lord knows I have a lot. So anyway, I submitted mine though. But I'll let you guys take it away.
All right. Thank you so much, Jessica. And yes, just a little preface. Thank you so much to everybody for who submitted questions. And if we don't get to them, in this presentation, I'm going to reach out to everyone individually who we don't get to cover those questions. So we will still be covering them, um, even if they're just individually. But thank you so much, and thank you so much for everyone for coming. So let's jump right in. So today, here's a little outline of what we're going to be covering in the next hour. So first, we're going to do a little intro to genetics, find out, what are genes, how can you read your gene report that you get when you go get sequencing. So next, Maheen's going to talk a little bit about the genetics specifically of FSASD or Salla disease, talking about inheritance and some common variants that we have in our community. Then we'll get a little Salla disease 101. So we'll learn a little bit about the biology underlying Salla disease. And next, we'll talk about the ongoing research. So what's going on um, with our FSASD researchers in their search for therapies. And two that we'll be talking about today are drug repurposing and then also genetic therapies. And finally, we're just going to close with some brief words about clinical trials. How do we get there? What do they look like? That sort of thing. Okay, so starting first with this little intro to genetics. So our bodies are made up of lots and lots of different things, these units called cells. And within a cell, there's this small compartment known as the nucleus that contains our DNA. So our DNA is wrapped up very tightly and contained inside this little compartment in all of our cells. So what is DNA? So DNA are the instructions within our cells that are then used to create proteins and other molecules that we need in our bodies. Now, when we talk about rare diseases, we often hear the word DNA, but also a lot of people mention genes. So then what is a gene? Well, if DNA is this entire strand, these entire set of instructions, a gene is then this small subunit, this small piece, small section. And usually we give each of these genes a name. And this particular gene would code for a very particular protein or molecule. For example, the gene of interest in Salla disease or FSASD is this SLC17A5. So if we zoom in and just look at this small section, this gene, we can see what the DNA is composed of. And we can start to see these instructions or this DNA code. And this code is composed of components called nucleotides, and they come in these 4 different flavors. Here we're denoting them as A, T, C, or G. Now, to go from the DNA code all the way to a protein, there are 2 different transformations that need to occur. So first, the DNA code is converted into RNA code, and then the RNA code is then used to create the protein. And the way it does this is by grouping together these bases in groups of 3 and signaling then which, in these groups of 3, signaling which piece or amino acid needs to be made or created to create this protein. So what happens when there are then variants in genes? So if we imagine that the DNA code is like a sentence, what would happen if we changed one of the letters? So here I have an example sentence. The sun was hot. We can read the sentence, we can understand it, but what happens if I change one of the letters? So here, the S in was changed to a Z. So I can still read the sentence, the sun was hot. It still makes sense, although there is a change. This type of mutation is called a silent mutation. And it's called this because it's usually benign, or meaning that it really doesn't have much of an effect on the function, the downstream function. So another mutation is called a substitution. So here in a substitution, we can change the S to a B. In this case, we can still read the sentence. So the bun was hot. We can still read it, but now it has a different meaning. And so this particular type of mutation could have a functional effect. A third type is called an insertion. Now, as I mentioned, the code is grouped into these sets of 3. So what happens if we insert a letter, here we've inserted an E, randomly in the sentence. So this E, which was placed before the sun, has now shifted all the other letters. And now when we look at the sentence, it no longer makes sense. And then this will likely have an effect on the function. So this mutation is called an insertion mutation. And the last mutation I just want to go over today is called a deletion mutation. So similar to the previous, if we would now remove a letter, again, we shift the code, and this leads to a sentence that we can no longer read or make sense of. So now that we know a little bit about the basic genetics that occur and what variants could look like, I'd just like to very briefly introduce how to read your variant from your gene report. So here is an example gene report. So when you get your gene report, there's a lot of information on there. There are many columns that are placed there. Here, I've only selected a few. But in general, there'll be a section telling you, about the gene, about the region of data, the region of that DNA that we're talking about here. If it has anything to do with a known disease, they will list that there as well. And then finally, if they find changes or a variant in that code, they're going to list that here as well. And any information on the functional outcome of that change. So whether it could lead to disease or not. So here I've listed the most common Salla or FSASD variant, and I'd just like to walk you through, what this annotation here means. So if we focus first on this part in bold here, the C115, C to T. So this part is talking about the variant at the level of the DNA, so that first level that I was talking about earlier. So the C here is what tells you that it's talking about the DNA code. The number, the 115, is telling you in that code, it's at position 115 that we're zooming in on, that we're talking about. The C here is saying that typically when you look at this gene, this region of the DNA, at spot 115, usually there's a C. And then the next part here, this caret to a T, is telling you that instead of the C, what they found is a T. So that is the change that they found. So now we're going to focus on the next part included here, this part in parentheses. So this part refers to the change at the level of the protein. So now going all the way to the protein. So this P tells you that it's talking about the protein sequence. The ARG here is telling you this is what is usually found. The 39, again, is talking about the position in the protein code. So at spot number 39, you typically find an ARG. And the last bit here, the cis, is telling you what the new amino acid is that they found there, the new piece of that protein. So instead of the ARG, they see a cis. And the ARG and the cis, these are just the names of those different components that make up the whole protein. So just to summarize this part of the talk, so the variant here is talking about one change, but when they list it on your gene report here, they're looking at it or calling it two different things, two different ways. So first, in the bold, is they're naming it by the change that they see at the DNA level. And the second is the change that they're seeing at the protein. Okay, now moving on, for the next part, Maheen is going to tell us a little bit about the inheritance of FSASD or Salla disease.
Okay, so for some disorders, well, I'll go back. All of us have two copies of each gene. Mom has two and dad has two. Oh, can I use my laser pointer, Sarah? Or you might
I
be the
think
only
I
one.
think it's just me. Yes, I can
Okay.
try to.
Yeah, you point at stuff as you see fit.
I'll try.
And mom and dad each pass one copy. Now, as you can see, let's say they have these four potential children. There's four combinations of ways that their copy of the gene could be passed along. So you see the unaffected son, he got one solid rectangle, and another solid rectangle from mom. And we'll say that this is maybe a variant or just a copy of the gene that doesn't have any changes that affect the eventual protein's function. The next two siblings, you see that we don't know which parent they got in this case, but they got at least one open rectangle and one full rectangle from a parent. And for autosomal recessive diseases like FSASD, that means that having one copy that could lead to a change isn't enough to cause the disease. Unlike other disorders, you might have heard of Huntington's. So in that case, getting one copy with a pathogenic or disease causing variant is enough to manifest symptoms. And so in this case, 50% of the children that they have would be carriers but unaffected. And then on the right, you see that this child by chance got two open rectangles. and they will have the disease. And so for recessive disorders like this, if you have a mom that's a carrier and a dad that's a carrier, 25% would have FSASD. And just to emphasize again, you have no control over which variants that are passed along to your children. It's all random chance. Um, and for disorders like this, being a carrier doesn't mean that it's hard to know if you didn't have sequencing or another family member that this runs in your family. You can go to the next slide, unless you have anything to add, Sarah. Okay, this looks really overwhelming, but kind of delving back into what Sarah was talking about, where you have this long DNA code, and then based on that code, you group 3 letters to make an amino acid that builds the protein. That protein strand has different parts of it. They fold in certain ways. They can make pores so that things can flow through. Maybe they help chaperone something that needs to cross from one place to another. Regardless, that means that changes, because they can happen anywhere in the DNA, can fall anywhere in the protein. And so these changes, kind of like Sarah talked about, can arise sporadically. We generate mutations, I think in the research they say like every new generation you yourself, your cell machinery makes 60 mutations or variants. And not all of these cause disease, but when they're pathogenic, what happens is over time, these can be passed down. And in the case of autosomal recessive diseases like FSASD, if you happen to have another parent that carries a pathogenic variant, those two can come together. And so that you present the disease in the child. The Salla disease variant, so the arginine to cysteine mutation, that's kind of highlighted in that red box, is the most common variant. Some of you maybe are a carrier for this, and you might wonder, but I'm not even Finnish. I'm not from Salla. Why do I have this? Well, like I said, these changes arise sporadically. So even in the first person in Salla, Finland, that got this variant, it just happened in their body. And because Finland, for example, or even Puerto Rico, they're islands, you don't have much people coming in or passing out, passing through. And so over time, as generations, continue forming, people, more and more people get that variant, which increases the likelihood that you will have it or pass it on. I think the only other thing is like, oh, no, you're okay. Like Sarah mentioned, there could be substitution variants where it changes the amino acid that's read. So that's, again, what the arginine to cysteine is. But as you can also see in a few places here, you'll see the word 176 del 1BP. So that tells you one letter was deleted. And so these changes obviously can be very varied. You can have deletions, you can have some letters inserted. But again, it's all through random chance that these happen. Jessica.
Thanks. And I find it very interesting that you say that every generation might be a new mutation, because I know that Michael's mutation has never been seen before.
Mm-hmm.
So even if they ran us against each other in a genetic screening before we had children, we would still not come up as a hit for Salla disease because his mutation has never been identified.
Yeah. And
that's fascinating that you say that.
Yeah, and that's actually a big issue with a lot of disorders. They call it missing genetic heritability because so many of these changes can happen, and for some of them, you might be the first person that has this change, and we don't know if it affects the protein's function. Maybe it leads to a different amino acid, and it's fine because for the overall protein coming together and its structure, it doesn't change much. But sometimes you can have a change where now the amino acid's negative and it's supposed to be next to something else that's negative. And so, like magnets, instead of attracting each other, they repel. And so you can imagine functionally that kind of affects how the protein folds. But yeah, Jessica, that's a great point. it's all by chance. And these are just the variants that we have seen and confirmed cases that we know are affiliated with disease, but there's probably way more um, in them. Some that are pathogenic and some that are not. Next slide. Okay, so Sarah mentioned you guys had some really, really good questions about the mechanism of FSASD. So I will try my best to answer these in a way that makes sense. But I would like to say I was incredibly impressed because I had to question my life after seeing these questions because I'd never thought about FSASD in this way before. Okay, you can go to the first one. So
Mhm.
in general, um, the idea of lysosomal storage isn't really well you might have not learned it in school. So the best way we like to talk about it is that just like in our human body, we have the digestive system where the food we eat gets chopped up into little pieces. We keep what we need and then excrete or poop the rest out. Lysosomes are an organelle. So tiny, that's it's like the same word for tiny organ. So baby organ in the cell. And their job is really important because they process waste or break things off as they need and either get rid of it as waste or they help keep other substances that can then be reused. So in the context of SLC17A5, this gene encodes silin, um, and silin's important because it forms this little door, I like to say, on the membrane of this lysosome so that silic acid that's inside of it can get out. And so you can see in the first picture, you have silic acid, which is purple, those purple little diamonds, and that core is letting them go out into the main body of the cell or the cytosol. But in FSCSD, there's two issues. So we find that, at least from the research, not only is maybe the folding or the function of that protein to enable things to pass through impaired, but you also don't get that core um, localizing to where it needs to be. So unlike in the healthy one, where maybe they have 5 of these silin transporters, in the FSCSD patient, maybe in this representative picture, there's less. And so not only is it impaired in their ability to get out, but there's fewer in general. Next slide. So one question was, is FSASD like diabetes? Because you probably keep hearing the word silic acid is a sugar. So when we say sugar, you're right. It is a carbohydrate, just like glucose. So I've shown the structure of glucose here. It has 6 carbons. Silic acid actually does derive from glucose. And over a series of steps, it gains carbons and becomes negatively charged. That's what that little minus sign is. But essentially, the mechanism of the disease is different. So, firstly, diabetes is an issue where glucose that we get from, eating food or, drinking sweet beverages, anything with energy, that glucose can't get inside of our cells. And because it stays in the blood, then you have literally high sugar in your blood, which then affects oxygen from getting to cells. And if you've ever heard about you need to keep you need food for nutrition and energy. That energy is coming from your body breaking down glucose by bringing it into the cell and putting it into a form that we can use to power everything else we need for daily living. And FSASD, this is an issue where silic acid can't get out of the lysosome. So this is all an issue inside of the cell. And in humans, especially, this is silic acid's not an energy source either. So even though it comes from glucose, it has a completely different responsibility to our body. Silic acid's really important because it tells when it's on certain proteins or maybe little fats, it tells them where to go. It's also important if you have two things that really need to interact with each other, then maybe sometimes silic acid is marked as like here, come, I'm right here, like, come find me. So they have completely different roles. So Good question, because I had to really think in my head, like how to compare the two. But if anyone has questions about this, let me know. But I hope that kind of explained a little bit of the difference. And so the next question is, would eating less sialic acid help? Also a great question because the issue is sialic acid storage inside of our cells. So first thing is that the way we get sialic acid, it's not really majority through our food. So even though I said we have glucose that we eat and is converted inside this little factory, I tried to represent it as a factory with the machinery inside of our cells, that process is always ongoing, right? So once that sialic acid's made and it's put onto things that need it so that those can get to where they need to go and do their job, they can come back, the sialic acid's cleaved or cut off, put into the lysosome, and then the lysosome's like, I'm going to let these out so we can reuse it. Or it'll destroy what it doesn't need left. So even if there was a way to avoid sialic acid, there really isn't, because your body will make it anyways. Um, your your body would make it anyways. It's really hard to eliminate, and also it's something we do need. So unlike other disorders, for example, in some diseases where patients can't break down a certain amino acid, you might tell them eat less protein because protein's made up of amino acids, and so maybe the one they can't digest is in there. But that wouldn't apply for this because um, like I said, sialic acid would form anyways. It's important, and it's not a breakdown issue. It's an issue of letting that sialic acid out. Um, yeah. Okay, so the next question was, is FSASD degenerative because sialic acid accumulates? So that's also a good question. And I like to compare it to another disorder I'm aware of, for example, phenylketonuria, right, where they can't break down phenylalanine, this amino acid. And in that case, we do know that high phenylalanine itself is toxic to the brain. In the case of FSASD, we need to do more research because we actually don't know a lot about how sialic acid levels change over the course of your life. So even if your kid has had their sialic acid levels measured in their urine or their CSF or blood, we don't actually translationally know what that means in terms of how it affects cells. Also, we're learning more about what silin does. Um, it also is responsible for things outside of sialic acid, perhaps, that we're learning more about, which would help. We do know that sialic acid levels are higher in patients who have more severe forms of the disease. And that's because bigger changes in that DNA sequence lead to bigger changes in the ability of that protein to function. So less sialic acid can get out. But in terms of the actual cause and effect, understanding that would be really important. So that's where you guys come in and our animal models to understand that.
Wonderful. Thank you, Maheen, for explaining all of that. So now we're going to move on to the next part where we get a little bit into the research that Maheen was hinting at. So some of our ongoing work in the FSASD consortium. So at the Star Foundation, we are very lucky to have this whole consortium of researchers studying FSASD and Salla disease, like Maheen here. And so here in this image, I just wanted to highlight, a few researchers that STAR has awarded research grants to. And their work in particular is focusing on uncovering some new therapies. And these researchers are approaching this question using several different strategies. So, what are the strategies that we can use for developing new therapies? So, typically, there are three different kind of strategies that one can use. One is gene therapy, another is drug repurposing, and the last one is creating novel small molecules or novel drugs. So the one that I'm going to be focusing on here is drug repurposing. So what is drug repurposing? So this is where we find already FDA approved drugs that have been used for other indications, other diseases, and we try to take them and see if they also work for our disease of interest. A classic example of this is with Rogaine. Many of you may have heard of Rogaine. So originally, this was supposed to be used for high blood pressure. However, researchers noticed that a side effect was hair growth, increased hair growth. And so in another, indication, another disease, male pattern baldness, this side effect would actually be really beneficial. And so this drug got repurposed for that use. So there are a lot of advantages for drug repurposing. One being reduced cost in getting this drug to the final stages where we can use it for another disease. So there's reduced time as well, reduced time to get it to patients. And there's also reduced risk. And this risk that I'm talking about is reduced risk of having a failure when you reach clinical trials. And all of this, the reduced cost, reduced time, reduced risk, is because these drugs have already done some testing in humans and in clinical trials. And so there already exists some data about the safety of these drugs. So seeing that they're not toxic at certain levels. And so because of this already available data, we could use this data to kind of skip some steps and go more quickly to our own clinical trials in a different disease. So that's kind of the idea of drug repurposing. And in our FSASD consortium, we use animal models to try and uncover novel therapies for FSASD or Salla disease. And two in particular that I want to highlight here are the zebrafish lines that we have, as well as the R39C mouse model. So there are several advantages to using these models. For the zebrafish, the advantage is that they are very easy to breed. So you can get a lot of different fish, do a lot of different experiments, and you can do these experiments rather quickly. So they're good for screening a lot of different drugs. So it's a nice model to try and find novel drugs to repurpose. With the mouse model, so they're closer to humans because they're mammals just like us. And with them, you're able to do more complex behavior. So you're able to look at things that maybe have more impact and more relevance to the disease you're studying. So recently we had the STAR Translational Research Award that went out. We awarded two. And one was to Greenwood Genetics, who's using these zebrafish lines. And another was to the NIH, who is using the mouse model here. And so the basic idea of this project is for Greenwood, they're testing a lot of different compounds, several different compounds that are already FDA approved. So trying to repurpose these compounds for FSASD or Salla. And what they're doing is they're treating these fish, and then they're doing some behavioral testing. So these Salla zebrafish have a particular swimming defect. So they swim a little differently than their wild type or typical developing zebrafish. And so what Greenwood is going to do is they're going to apply this drugs and try to see if they can correct the swimming of these fish, see if it has a beneficial effect. At the NIH, they are, oh, sorry, Agnes, do you have a question? Yes.
So Sarah, like with you're testing they're testing drugs on fish. So that means that there's literally a drug like one of us might take, like through a pill in our mouths, but they'll put the drug in the water and then the fish swims slower or faster, or like
It
how do they know if a drug works on a fish?
Yes, so they have the fish sort of in these little plates. And so you can see they're in different wells, you can see a different fish in each of these little wells. And I believe for the zebrafish, for the drug application, I believe it is directly just into their water that they're swimming in. I'll have to look that up to be certain. I haven't worked with fish myself, but yes, they will administer this drug and then they will track the movement of the fish. So they have some sort of imaging tracking software. So they're able to watch how the fish swim. And it's just a particular movement that they're doing differently than the other fish. And so they're measuring this difference. They can see a difference. And then they're trying to see if they give the drug, do they start looking more like the wild type or the typically developing fish.
I think at some point Heather mentioned that the mice without like lacking the protein, they swim in a circle
That's
instead
what
of
I think I have seen an
going
image
straight.
of that as
Yeah.
well.
So their path is more like squiggly instead of, having a direct like path to where they want to swim.
That's really that's pretty neat actually that they can use it. So basically there's fish that have the solid disease gene deficit, and then there's fish that don't. And
Mhm.
so then they look at the fish that does versus the fish that doesn't and see how they're different. And so then they
Yes,
can tell if the drug works.
yes. And these fish, so they're very easy to breed, so you can get a lot of different fish, and you could put a
Mhm.
lot in that plate, and you could test many, many different compounds. And so it's a really nice tool. Heather always talks about the utility of this, that is really nice to be able to screen many different drugs. And so if there's anything that comes up that the researchers in this consortium are interested in, hey, maybe this is something that could help. This is a really good model to be able, as a first pass, to test that. And
Okay.
then they'll take the data from that and test it in the mouse later. That's that's
Got
an
it.
idea. Jessica,
Thank you. That's a great explanation.
Jessica, did you have a question?
How do you give the fish um solid disease? I know that the mice existed. They were existing in a colony somewhere that they had solid. How do you make sure that they have solid disease?
So
Is that a
that
dumb? Sorry, that might
no,
be a
that's not
dumb
that's not
question.
dumb at all. It's just a little
Okay.
above me. I'm not but good on the genetics. But it's a genetic model. So I don't know, Maheen, if you have
Yeah.
a better, maybe you're a better grasp on the
Yes.
genetics.
Yeah, because this is the perfect tie-in to the question about genetic therapies. So, um, with at least for the mouse model, so you're right. It just like humans, where we could randomly have generated a variant that causes disease, like the Arthur9C, we, at least the mice that we made at the NIH, we genetically edited them to make sure they had the specific change. And so we used CRISPR, if people have heard of that. And with CRISPR, what you can do is, I wish I had a good picture, but DNA has two strands and they look like this. And let's say the variant or the base, the letter that I want to change is right where my, whatever you call that, my little knuckle is. So you have these molecular scissors that have a GPS with them, right? They are only supposed to cut where they're supposed to cut. And they know where they're supposed to cut because there's a sequence very specific to this part of the gene. So let's say SLC17A5 that makes it highly likely to go here and not to randomly cut anywhere else in your genome. And then once it cuts, you can either insert another base. So in this case, It's a, like Sarah said, it was a C changed to a T at position 115. And so that's what we did because the mouse sequence for SLC1785 is the exact, very, very similar. I think there's only a couple letters that are different, but at this position in particular, they both have a C. And when you change them to a T, they have the same amino acid difference. So it changes from arginine to cysteine. And so, yeah, you just use these molecular scissors, they cut on both ends, you insert what you want, and then your body has repair mechanisms and glues it back together. And so it'll read properly now with that change that you wanted in there. You can do the same thing if you, instead of substituting it, you wanted to insert something. So you cut, insert a bunch of letters, and then your body glues them together. And now it has one consistent sequence. And they did the same thing with the zebrafish because they also have a version of SLC17A5. You might hear the word homology. So it just means that across certain species, certain genes are very important. And in this case, SLC17A5 is likely very important. It's probably smaller in zebrafish, so that's what we'll see across certain species, that the overall protein or maybe the DNA code is slightly different in length, but they tend to have a similar function. Did that help answer the question?
Yes.
And just
My niece is going through this right now in her AP biology class as a senior. She's going to go ahead and be a
Yeah,
nurse. And it's the first year that they're talking about CRISPR technology.
yeah,
And she called me the other day and she said, "What? But when you said there's a GPS attached to that little scissors,
yeah,
that makes so much sense because then it's following the map of the DNA
exactly.
and it
Yeah,
knows where to cut because of the GPS."
yeah.
I always wondered that.
Yeah,
Okay,
it tells it
it's
exactly where to
fascinating.
go. But that's the other issue, right? You have to make sure it's very specific for where you want it to cut because it's possible it'll cut in other genes and you don't want it to do that. And so it takes a lot of time to develop like the little map for these like scissors to follow.
wonderful. Thank you so much, Maheen, for that wonderful answer. Okay, so yes, so we have these lovely zebrafish and mouse models, and the current work, yes, is using them to test these repurposed drugs. With the mouse, they're doing a little bit more. So they're also looking at brain health and if there's anything that they could use as a biomarker. And a biomarker being something that you could measure over time to see how the disease is progressing. And then to see if we treat, does that biomarker change? And in addition, they're also doing some behavioral testing that would be relevant to our Salla community, looking at motor function, which we hear about a lot in our Salla community, um, looking at learning, and then also survival. And they're looking at all those things following the drug treatment. So, how could the clemistine, just as an example, help with SALA or FSASD at the molecular and cellular level? Like, what is the idea there? So, the basic idea is that in our brains, we have these cells called neurons. Here I'm showing one in purple. Um, these cells communicate with other neurons through these electrochemical signals. So ions or charge is sent down their processes and then delivered to the next neuron. So they're sending these signals to one another. Now, to make this transfer of signal efficient and fast, there's also this additional cell type called an oligodendrocyte, shown here in blue. And what this oligodendrocyte does is it comes with its feet and wraps around these processes, insulating them to make the signal go down faster, make the signal more efficient. So this wrapping you've probably heard about is called myelin. So this is actually the feet of that oligodendrocyte wrapping around. Here I'm showing this cross section. So the purple is that neuron, the arm of the neuron, and the blue is the foot of that oligodendrocyte, that other cell that is just wrapping around that neuronal process. And you can think of this wrapping, this insulation, as like the wrapping or insulation of an electrical wire. So it's something that is helping the signal go more efficiently and go faster. So in a typical neuron, a typical case, we have a set number of these oligodendrocytes that are wrapping and a certain amount of myelin. And what's been found is that in Salla or in FSAFD neurons, there seemed to be this decrease in myelin and decrease in these oligodendrocyte cells. And so the idea behind this drug treatment is that if we give this drug, perhaps it can help increase the number of these oligodendrocytes, increase the amount of myelin, the amount of wrapping, and make these Salla neurons look more like the typical neurons. So kind of return them to a more typical fashion and help with the signal that they're sending to one another. And that downstream that might have some beneficial effects on things like motor function, cognition, and even they're looking at survival. So that is the basic idea behind those drug repurposing strategies. So now we're going to hear a little bit more about these genetic therapies that we touched on a little bit from Maheen. So we're going to talk about gene therapy.
Alrighty. So, like I said, and some of you might have heard that whether it's for the FSASD community or just in science in general, that CRISPR gene editing has been very big. I actually don't know how long it's been around, but it's a very good genetic tool for creating models for different diseases, especially our FSASD models. But the idea is that if we can manipulate DNA like that, then maybe it's also a way to correct changes that lead to disease. So, like I mentioned, it's basically I like to imagine it as a pair of scissors with a GPS. They recognize sequences that hopefully are very specific to where they need to go because there's a lot. If you imagine how very, very, very long your DNA code likely is to make up everything that we are, there's lots of parts that look very similar, even though they're supposed to encode different things. So a lot of time and effort goes towards making sure that this GPS and this map is going to make it go exactly where it wants, with very minimal, if not none, going to another gene or area in the genome. So traditional CRISPR, if you can see the DNA has two strands, and like I said, these bases here are kind of attracted to their partner, like this. CRISPR, whether you want to add bases, delete something, or switch something out, it has to cut both the top and the bottom strand. Insert whatever change, and then relies on your body to glue them back together. So if you can imagine, while this sometimes is successful, there's a lot of risk with it because you're relying on a process of your repair machinery to fix this genetic code. Base editing, which you might have heard Dr. Wang's group is very successful at performing in a wide variety of animal and cell models for diseases including FSASD. It's a gentler version. So you still have, the molecular scissors and they're still using their GPS. But instead of cutting both strands, I like to think of it as like an eraser. So because only one strand is the coding strand, that's why in your gene reports it said C dot maybe what 150. How does it say on their gene reports? Yeah, it's like C dot
115.
exactly,
Mm-hmm,
like
mm-hmm.
115 C to T. So we could go exactly to base number 115 on the strand that we know actually makes the protein, because the other strand is non-coding. They just need to be structurally sound, so they have this partner. But let's say only this one is going to make the protein. These molecular erasers come in, go to exactly the base they want, erase it, and insert the correct base. And so this one is a lot safer because you're not cutting, you're not especially affecting both strands. You only need to affect the one. And they, at least at Chalk, they used this machinery in a cell model that had the arginine to cysteine mutation, or like I said, the C dot 115 C to T. And they showed that after using this machinery to correct the T back to a C, um, they saw that cells actually had DNA. They still had some altered, there were still some cells that, didn't have complete correction, but in this petri dish, a lot of them did. And so the idea is, since in cells, this has been, you know, promising, and now that we have a mouse model that is compatible with this base editing, so again, because it's only editing one base, it's only suitable for mutations that impact one base, if that makes sense. So you wouldn't be able to correct something where 10 bases are missing with a single base edit. It wouldn't do that. For that, you would need traditional CRISPR because you want to insert a bigger sequence versus erasing a single base and putting the right one in. But since we have the mouse model, we can try delivering that, seeing where it goes and if it can improve the mouse's phenotype. That is their motor behavior, what their brains look like, just how long, what their motor behavior looks like in general. Because we know over time, at least with my experience with the mouse model, they progressively become ataxic. So while they're a little wobbly at 3 weeks old, by the time they're 5 to 6 months, while they still move around, you can tell that their legs are significantly more unstable as they move. And so something like that would be an easy way to see, oh, they're actually walking a lot better. And we also have healthy mice, so they're healthy siblings that don't have FSCD around. So you can visually compare with them too. And the idea is also we can look at their urinary free sialic acid levels too, which we have done. And we also know that the mice have compared the mice with the Salla variant compared to their wild type or carrier siblings, which both are healthy. They have significantly increased free sialic acid, which we also see in our kiddos with FSASD. So that's also something you could look to see if it is improved if this base editing works. Um, any other questions? Sarah, I don't know if you have anything to add.
No, I think that's pretty good on this part. Okay, so we're getting to the end here, so I just quickly will go over a little bit about clinical trials. Just very brief kind of information about what those steps kind of look like to get there, to get a drug onto the market. So the first step is kind of what we're doing now, this research and proof of concept. So you figure out molecular pathways of the disease, you figure out the drug and how the drug would work, if it's safe, and what dosage you would give in an animal. And next, you interact with the FDA. There's two kind of meetings that you can have with them. The first is where you get kind of information and advice for them on how you would move forward to get to a clinical trial. You give them the experiments you've done, you talk to them and see how could we make this better? How could we make sure that you're going to approve us for the next section, for the next step? And then you actually have that official meeting with them in that application. And there are some things that you need to gather in order to have this application. You need to have some safety information. You need to have a plan for how the drug gets manufactured, how you're going to give the drug. Is it a pill? Is it an injection? And then also these clinical endpoints or biomarkers that we've kind of been talking about that could be For us, it could be sialic acid. Maybe that's something that we measure over time, and maybe that will change it, upon drug application and drug treatment. And so these are the kind of things that we need to gather in order to then move and get approved to have a clinical trial. And the clinical trial has three different phases. First is where we assess the safety. And so this is just done in typical humans, typical people, just to see if the drug, can you tolerate the drug? Is there any toxicity? Then you can move on to testing it actually in the population you want. And then on the third trial, you kind of expand the age range and the number of people that you test this on. And all of this really focused on making sure that what you're doing is safe and the dose you're using is safe and the amount of time that you're going to be treating is safe to do that. And so that's what the FDA, that's what their goal is. They want to make sure that they're protecting all of our patients, all of our community. After that, if you have a successful clinical trial, then you can apply again to the FDA to say, we had a successful trial, please approve this drug so that it can then be used and prescribed. And that is when, if you get approval, you can start manufacturing and people can start prescribing. Now, as I mentioned, we're kind of on this beginning phases. So we have a lot of this research and proof of concept going on, and we're starting to kind of think about and collect things that would be in this package that would eventually go to the FDA. So thinking about things like clinical endpoints and biomarkers, we're looking at those things in our mouse model, in the zebrafish model, and starting to think about these things and how we would get to them. Now, I also mentioned that with drug repurposing, something beneficial there is that maybe we could skip some of these steps, as in we wouldn't have to get the safety information or the route of administration. We wouldn't have to come up with that fresh and new because there are already clinical trials of clomiphene going on. There are several different, one for optic neuritis, Williams syndrome, multiple sclerosis, and even depression. And so there's a lot of information out there that's already being collected on the safety and toxicity and the different dose that could be used. And so we might be able to piggyback on this and use some of that data to help us move forward and move faster. And that is one of those advantages of doing this drug repurposing. So then just to end, I just want to talk about, so getting ready for the clinical trial. Again, so this safety information, the route of administration, those things, hopefully we would be able to get from these ongoing clinical trials and kind of apply them to a case with Salla. And what we need to next be focusing on and what we're starting to think about is what endpoints and biomarkers that we would use in our community. And the way that we get this information is through our natural history study and through biobanking. And so this helps us figure out what needs to be measured, what we can measure over time, and what we can see if a drug is helping, things that we can measure to try and see those differences and those changes, and show the FDA that this is something that works for our community. And so with that, I'll just wrap up. Thank you, everyone, for your attention, for your questions. Thank you to Maheen for joining me here today. And if we went a little over, but if anyone has any more questions, we're happy to take them. And of course, you can always email, and I will be very happy, and I'm sure Maheen will as well, to answer any more questions that you have. And oh, I don't have Maheen's email up here, but we can get
I
that
can
out. We
drop
can
it
get
in
that
the
out.
chat too.
We can get that out to you, yes. But yes, okay, we have a question.
Yeah.
Alyssa?
Sarah, can you take down the slides?
Oh,
Thank
absolutely.
you.
There we go.
Okay. I just had a quick question about, so we talked about like the oligodendrocytes in terms of like hypomyelination, but are like the Schwann cells also affected or is it just in like the central nervous system?
Oh, that is a good question. Maheen, I don't know if you know that part. I would
A
assume
great
that they
question.
are affected.
So it's one of those things where we need to do more research. So, for example, wow, I love that you brought up Schwann cells. I'm so excited. So, for context for Alyssa's question, like Sarah mentioned, the oligodendrocytes are the primary cells that make myelin for your brain and spinal cord, but we still have nerves throughout our body. And so, for what you call the peripheral nervous system, meaning everything that's not your brain and spinal cord, they use Schwann cells, but they make myelin. When, at least in the mouse model, when we looked at their sciatic nerve, which is a peripheral nerve, to see if they had myelination issues, we didn't really see any significant changes in terms of that.
Okay.
Obviously, that doesn't mean there isn't a deficit because it is a genetic disorder. So you'd assume that the change is there. We just don't know how important silent function is in some cells over others. So clearly, it's very important for oligodendrocytes.
Mm-hmm.
It could also be that the time point, like the age at which we checked these mice, wasn't old enough for us to see those changes. At least in some of the patient clinical studies, and Sarah, correct me if I'm wrong, it's not very consistent, for example, in the presentation of like peripheral neuropathy or like on nerve conduction studies, that there's many changes in that in the patients. I know it's probably been mentioned like one or two, a couple cases, right? But not the overwhelming majority. Whereas, for example, the brain myelination defect, all of the patients have that. So
Yeah.
um, it's something we definitely are interested in looking into. Um, and I'm trying to look into, but we don't have a confident answer right now.
Yeah, thank you. Thank you for that answer.
Yeah,
That's
awesome
makes a lot of
question.
Thank you so much, Sarah. Does anyone else have any, it's a lot of information to drink in, and this is really a starting point, for our kind of solid disease education, and particularly with, with the research where it's at right now. But does anyone else have any other initial questions or basic